All right, guys. So we're going to get started on our discussion about receptor tyrosine kinases. These are a family of plasma membrane receptors with protein kinase activity. They're going to transduce extracellular signals by a mechanism that is fundamentally different from that of GPCRs, G protein coupled receptors, which I believe you guys talked about already. But these are going to be a little bit different. And the key here that I need you guys to focus on is, yes, the pathways, of course, are important. You're going to have to know the pathways, but also the subtleties that are associated with receptor tyrosine kinases, or we also like to call them RTKs. RTKs, if you see that, we're talking about tyrosine kinases, right? So, again, these are going to have a little bit of a different mechanism, or actually quite a different mechanism from GPCRs. We're going to see a little bit of trans-autophosphorylation. We're going to see catalytic domains. We're going to see all kinds of different messenger systems going on to give us that final reaction that we're looking for. And what we mentioned in the beginning, in the first part of the session during the live review, was that we kind of went through the nomenclature aspects of things, right? And Dr. Larea really emphasized nomenclature in his lecture because it is important, and it's going to really help you guys to truly grasp the subsequent aspects of the lecture. Okay, so we went through those. Go through those if you haven't already. But this is going to focus more on actual pathways, right? We're going to actually go through the step 1, 2, and 3, and 4, and I'll explain the little nuances as we go. But this is just going to really be more of a comprehensive overview of the actual steps that we're looking at. Okay, we'll also be covering blood coag as well. So we're just going to start here and finish up our cell signaling because it is a big topic, and then we'll jump right into blood. Okay, guys, let's get started. So the first thing that we want to remember about our receptor tyrosine kinases is that they have a ligand binding domain on the extracellular face of the plasma membrane, right? So in our discussion live, we drew out this picture, right? And we showed over here the extracellular portion. We had the membrane drawn over here, right? And then we had the transmembrane aspect, the transmembrane site. And then over here we had the catalytic domain, this red part over here. And it's important that we realize that because the whole thing is quite separate from our discussion on GPCRs, which we remember had that that serpentine, that's those seven transmembrane domains, right? And they were all connected in their own way, right? 1, 2, 2, 2, 2. They're all connected. You have your interior portion, your exterior portion. This is different, right? So the receptor portion is going to be out here for receptor tyrosine kinases. And they're going to be connected by this single transmembrane segment right here, the purple in the middle. And we're going to use insulin a lot as just kind of the basis upon which of a lot of our continued examples will follow, right? It's going to be the main example because we know a lot about it. We know a lot about insulin. Like with GPCRs, the textbook likes to use adrenergic receptors, right? The alpha 1, beta 1, alpha 2, beta 2 receptors. For those of you who read the textbook, it's pretty largely, it's the center upon which the entire GPCR topic is built. But we know pharmacologically, we know a lot about insulin, so we're going to use that a lot. In fact, the receptors for insulin and also epidermal growth factor are going to be the prototypes for, I believe, 58 different RTKs in humans. So we're going to use insulin, the insulin substrate, as kind of our way of turning this into a real world example. Okay, so let's start off with the stimulation of an insulin receptor. Okay, so we're going to start with an insulin receptor. We're going to think of this tyrosine kinase receptor as an insulin receptor. Okay, and that means what? Insulin will be the substrate. All right, guys. So let's think about the breakdown, or rather the individual components of this RTK. Right. So here's actually a good picture they give you in the book. Right. This is inactive. Right. We don't see any ligand. There's no ligand there. Okay, and this is inactive. Notice the positioning of these two subunits, right? They're separate, they're far away. These ones are close and intimate, they're really close, and that is for a reason. If you notice up here, we do have our insulin substrate, we have that ligand readily visible, right? Okay, so that bound. Now, insulin receptors in particular, and this is specific to insulin, but it might help you understand the general idea, right, behind RTKs in general. So, in insulin, there are two subunits I want you to be aware of, right? In insulin receptors, excuse me. Okay, so there are the portions outside of the cell, right? So here's our membrane. Here's our interior of the cell, right? So our intracellular portion. And here's our extracellular. This is the extracellular aspect, okay? The insulin binding region, right? So here's a little insulin. Insulin is going to come in here and it's going to bind to this region. That insulin binding region of the insulin receptor lies outside of the cell, this portion, and comprises of two intertwined alpha subunits. So here's an alpha subunit and here's an alpha subunit, right? The binding of a single molecule of insulin. So once this insulin binds, it comes in. That binding, because these two are intertwined, right? So think of these two as a unit. Once it's bound, once that insulin binds, it is going to be communicated, right? So this part over here, all of this is going to tell the rest of it, is going to tell the rest of the receptor. How? Through these parts, through this transmembrane aspect, right? And that is going to be called a beta. These are our beta subunits. Okay, so here's beta 1, beta 2. Again, don't get too bogged down with the alphas and the betas. It doesn't matter. Okay, this is just specifically to insulin, but it should help give you a specific idea of what's happening here. Okay, so insulin binds. These receptor portions, the alpha subunits, they recognize it. Okay, that information, the fact that insulin is now bound, is told to the rest of this. How? Through the beta subunits, the transmembrane portion. Transmembrane helix of each is going to pretty much tell the rest of this. Okay, now we have, hey guys, insulin came in, the alpha told us, okay, so now these guys know we have insulin, we have the receptor, it's time to get to work. Okay, great. At this point, once the insulin is bound, these two are going to know it's time to come together. It's time to come together. Okay, so once they come together, that's when you look into this. That's when you're seeing this intertwined aspect over here. You see how these two are connected now? They're going to phosphorylate each other. Remember the first video we talked about that trans autophosphorylation? That's where this is going to happen. Okay, so once these two come together, these two beta subunits come together upon being told to, right, by the binding of the insulin, the alphas basically tell the betas, okay guys, time to come together. They come together. Now, the tyrosine kinase domains inside the cell, these ones, these are your tyrosine, these are your catalytic. Remember we had in this picture over here? That's this part. These parts, these two little lobes at the bottom are going to trans-autophosphorylate. Okay, they're going to phosphorylate each other on 3 different tyrosine residues. 3 different tyrosine residues in the inactive form of the tyrosine kinase domain over here, right? None of these tyrosine residues are phosphorylated. There are tyrosines. Yeah, there's tyrosines all over the place, but none of them are phosphorylated. Once we get that insulin receptor activated, telling these guys, the catalytic aspect, okay, now it's time to get to work, then, and only then, will the kinase activity aspects phosphorylate the other on 3 different tyrosines. Okay? And the same thing will happen is this one will phosphorylate, each kinase part will phosphorylate 3 tyrosines. Right? Okay, so we got the autophosphorylation part down. All right. And that is pretty much step 1, guys. So here's step 1. Step one is basically just the insulin receptor binds insulin, undergoes autophosphorylation on its carboxy terminal tyrosine residues, right, which we discussed. Now, this whole image here, right, this whole thing is just a small portion of a much bigger diagram. And what I'm going to do is I'm going to take here step one, and then we're going to do step two, step three, and then we're going to slowly build on this one diagram to until we get the whole diagram, until we get the whole image, and you guys will be able to understand step by step exactly what we're talking about when you look at the whole picture. And that's the goal here. So first things first, I'm going to try and tie in a few of Dr. Larea's slides because they do make a lot of important points. So the first pathway, this pathway that we're talking about, right? We went through the trans autophosphorylation aspect, right? And we're using the insulin receptor as simply an example, as a prototype, that and the EGRF, I believe, epidermal growth factor, EGF receptor. That's going to be the prototypes, right? That we use kind of to teach these type of pathways. So now the first thing we're going to do from this point is we're going to look at what's called the MAPK pathway, right? The MAPK pathway. And that's going to be right over here where Dr. Larea laid out a lot of the important components of this pathway. That's the mitogen activated protein kinase pathway. Okay, so it's going to require a mitogen activation. We'll discuss all of these little aspects more and more as we go. But first, let's just go through some of the main discussion points, right? Some of the main components that we're going to see in this MAPK pathway. Okay, so here, yes, receptor tyrosine kinases. We got that down. What are some of the main pieces? Okay, RAS. This is going to be important. RAS is the monomeric GTP binding protein. Okay, so remember, maybe in GPCRs we had that heterotrimeric with the alpha, beta, gamma subunits. This is kind of the equivalent to that, but here we have a monomeric GTP binding protein. So RAS, that's going to obviously be somewhere closer to the beginning, as we see over here, right toward the front part, right toward the beginning. Okay, RAS, great. What else do we got? Oh, I recognize this. GEF, guanine nucleotide exchange factor. The guanine nucleotide exchange factor. Guanine nucleotide being what? Our GDPs, our GTPs, right? And we know that we're going to have to switch them in and out to activate or inactivate certain aspects of receptors. So, okay, so the GEF, the guanine nucleotide exchange factor, in the case of tyrosine receptors, right? Receptor tyrosine kinases, RTKs, we're going to be using GRB2 SOS complexes as our GEF, as our guanine nucleotide exchange factor. These two are two separate things, but they work together. So we'll get into that as well. RAF, okay, all right, we're moving down the line here. So that's the next thing that we see in there. RAF, the RAF is our protein serine threonine kinase. Okay, we talked about this. We talked about the different serine threonine kinases. We talked about how they're distinct but related to the tyrosine receptor kinases, right? And non-receptor tyrosine kinases. And we'll talk about the difference as we move forward. But RAF, okay, RAF. So we have RAS and RAF. These are two different things. MEK, what is MEK? Okay, MEK. That is basically just a little, kind of like, remember we had Janus, we had the Janus kinase, JA kinase, and that just became JAK. Right. Again, it's important. I'm highlighting nomenclature here because that is a major aspect of this whole discussion. Right. So yeah, Janus and kinase become JAK, J-A-K, JAK. Here we have, instead of that, we have the MAPK / ERK kinase, M-E-K, MEK, M-E-K. And that is going to be we talked about how there's tyrosine kinases, how there's serine threonine kinases, and sometimes there are dual specific kinases, and those are going to do both. Those are going to have active sites. They're going to be able to, two different active sites, ones that will phosphorylate tyrosine and others that will phosphorylate serine and threonine. Right? So this MEK is going to be a dual specific kinase. All right. Very important. We're going to keep that in mind. Okay, and again, this is just showing us, MAPK, the whole pathway that we're looking at, what it stands for. So mitogen activated protein kinase, and then ERK, extracellular regulated kinase, MAPK, ERK. A lot of times you'll see them; they'll be mentioned together, right? Okay, and look at this, important. We talked about this in great detail, at great length, right? The SH2 and SH3 domains. very, very important, SH2 domains and the SH3 domains. Sarc homology 2, Sarc homology 3. We know that the kinase, the initial kinase, is going to be our SH1 domain, but we never say that. We say kinase. We don't say SH1. We say kinase. But here, based on that Sarc prototype that we were talking about, we're going to have SH2 and SH3 domains. And that SH2 domain is going to bind our phosphotyrosines. Important, keep that in mind. We're going to talk more about that as we move forward. And this SH3 domain is going to bind proline-rich moieties, right? Proline-rich areas, domains, spots. Remember, domain is a spot. It's a place. It's a sequence, right? Okay, terrific. So now we know what the main players are going to be over here, right? We know what the main components are going to be, and that's part of the MAPK pathway, the mitogen activated protein kinase pathway, right? And again, as we always say, as we said in the last one, this is all gonna, yes, there's quite a few steps that we're using on this big orange arrow over here. Is, there's a lot of little steps that go into it, but at the end of the day, here is our focus. Here's what we care about: metabolism, differentiation, proliferation, survival. We discussed the definitions of all those in the first video, right? Now, that becomes a problem when we have unregulated metabolism, differentiation, proliferation, and then apoptosis, which is, programmed cell death. That's when we have a problem, when it's unregulated, right? That's when we have a proto-oncogene becoming an oncogene, which is problematic. We can't control it. It's unregulated, right? So we're going to talk about the MAPK pathway, like I said, step by step, and then we'll discuss the other two that Dr. Larrea really wanted us to focus on. Okay? So now the way that Dr. Larrea approached this pathway, right? And normally people will just try to memorize, and I know I said that, it is largely memorization. And while that is true, Dr. Larrea took a very intuitive approach by mentioning the hypotheses, by going through the different steps through which the researchers who made these discoveries, their thought process, right? And that'll kind of, hopefully help clear up the way that you approach these type of pathways, right? If you have a bit more of a background understanding, that'll be useful. Okay, so right here, I'm gonna jump a little bit back and forth between the insulin receptor and the epidermal growth factor receptor, and they're relatively similar. We'll go through the minor differences between the two, but the concepts remain, right? So over here, the title is, why is it commonly referred to as the MAPK signaling pathway? Okay, so first thing we see here is this: mitogen/ligand/primary messenger. Okay, we know that MAPK, as we said before, stands for mitogen activated protein kinase. Okay, mitogen activated. So this is the mitogen. Oh, okay, so the mitogen is just the ligand. It's the primary messenger. You know how we have secondary messengers, right? And now, so this is a primary messenger. This is the first one that's coming in. So this is an EGF molecule, because we're looking at an EGF receptor. And again, EGF receptors, insulin receptors, these are simply examples, prototypical examples of tyrosine kinase receptors, right? So we know, upon binding, we have the dimerization of these two monomers. Here's just a monomer by itself. And then when you have binding, we have these two monomers come together. They come together and become a dimer, a homodimer, because we have two of the same thing coming together. Okay, excellent. So what happened here? The whole point of this slide, the discussion is that, okay, we saw this all happening. We know this is a tyrosine receptor. But how come when they looked at the S6 protein, right? The S6 protein, S6 kinase, they just use that as their specimen for this experiment. They noticed that after the autophosphorylation, after the mitogens bind and cause this whole autophosphorylation reaction to occur, that ligand binding leading to dimerization, which we know that makes sense, and autophosphorylation on tyrosine, we know that makes sense. But it also led to serine threonine phosphorylation? Does that make sense? No, it doesn't make sense. Why would a tyrosine kinase phosphorylate serine and threonine residues? That doesn't add up, right? And that is exactly right. It doesn't add up. So what does that mean, right? Because they saw all of these. They saw these serine residues here. They saw these serine residues phosphorylated after all of this stuff happened. After this whole phosphorylation process happened, it didn't make sense. They didn't get why. Okay, so they have to come up with a hypothesis. They have to come up with an explanation, a potential explanation that they can test to see if it's true or not. A hypothesis, right? We all know what a hypothesis is. We talked about that since grade school. Okay, so what's their hypothesis? How do they plan to explain the phosphorylation of these little serine residues when we all we've done is use a tyrosine kinase? How do you explain that? Well, okay, they said the S6 kinase must be directly activated by the receptor. Does that make sense? No, of course that doesn't make sense. No, because the receptor, if it was directly activated by the receptor, we know the receptor is a tyrosine kinase. So that doesn't make sense because we see serine residues here being phosphorylated. Now this is the S6 protein. Okay, notice we're saying protein, that's different. So they decided, okay, their thinking process was along the lines of, okay, if we have a protein with phosphorylated serines, like we see in the S6 protein, there must be an associated kinase. And that's exactly what they moved forward with. They isolated an S6 kinase. That's what they called it. So this S6 kinase, they assumed that the S6 kinase must be directly activated by the receptor, right? So were they thinking the receptor turned on the S6 kinase, right? The receptor turned it on. How do we turn things on? We phosphorylate, right? And then they thought that the S6 kinase phosphorylated the S6 proteins. But here's the problem again. Same problem. The S6 kinase also had serine residues that were phosphorylated, not tyrosine residues. Not tyrosine residues. So that makes this hypothesis moot. The S6 kinase must be directly activated by the receptor. No, it must not be. Wrong. Because the tyrosine receptor is still a tyrosine receptor. Right? Here in the first hypothesis, they said that it must be activated. The S6 protein, they thought it must be activated. There's this big question mark. They thought it must be activated or phosphorylated by the receptor. Wrong. So their next hypothesis is, okay, here we isolated the S6 kinase, and we know that this S6 kinase is phosphorylating these serines on the S6 protein. But what is turning on? What is phosphorylating the S6 kinase? They'll hypothesize, hey, maybe it's the receptor. No, it can't be. Why? Because these serines were phosphorylated. So it has to be something else. There must be something else that is phosphorylating the S6 kinase. Okay, so you're telling me that there has to be a kinase for the kinase? Absolutely, I am. Yes, there's an S6 kinase, an S6 kinase. Yeah, that's kind of what they called it, right? And there were two groups actually working on this at the time, right? And they came up with two important names, two names that I hope you'll recognize, right? Let me go ahead and paste this here. Bear with me. Paste image. There we go. Okay. Oh, okay. Here's that S6 kinase. Let's just go through and make sure we're all oriented. Here's our receptor, right? Our activated receptor. Why do we know it's activated? Dimerized. We also know that there's phosphorylation happening on these tyrosines, right? Auto phosphorylation. They phosphorylate each other. Great. Okay. S6 protein. What we saw downstream, we saw, okay, this protein is phosphorylated on serines. Okay, what's phosphorylating the serines? Could it be the S6 kinase? No, yeah, it is actually. It's the S6 kinase. Okay, so they asked first, is the receptor phosphorylating these serines? No, because this is a tyrosine kinase. So it can't be that. That's what made them think, okay, let's figure out what what's doing the phosphorylating here. And they figured it out. They figured out it's the S6 kinase. The S6 kinase is responsible for making this happen. It's responsible for phosphorylating the serines. Okay, great. So maybe the receptor, this tyrosine receptor, is activating the S6 kinase. Nope. No, it's not. Wrong. How do we know? Because the S6 kinase also had serine phosphorylated on it. Okay, so that's not the case. So that means something else must be phosphorylating this kinase, and that's where we get the S6 kinase. Oh my God, there's a S6 kinase. Yeah, there is. Okay, so we have the S6 kinase. So this is a phosphorylating agent of a kinase. Excellent. And there were two people actually, two labs doing doing research on this at the same time, and one lab, identified it as what's called a mitogen activated kinase, or as we know, it's called the mitogen activated protein kinase, aka MAPK. Okay, it's all tying together now. Why is it commonly referred to as the MAPK pathway? All right, now it makes sense. I'm seeing where it's coming from. MAPK, mitogen activated protein kinase. Okay, so that is the S6 kinase. Makes sense. Got it. Extracellular, and the other lab that was doing the work, they identified it as the extracellular regulated kinase. Okay, ERK. We're familiar with that. MAPK, ERK pathway. Okay, so interesting. We're going to keep that in mind. So we know that about the ERK. The ERK here, that's simply just a member of the MAPK family. Okay, the ERK, the extracellular regulated kinase, is just a member of the MAPK family. We'll see that in the next slide. All right, but so let's take a look over here at this sequence. Okay, the S6 kinase. Is it possible that maybe this S6 kinase is being activated by the receptor? Because look at this. Here we have a threonine and a tyrosine being phosphorylated. And they thought, maybe the receptor finally did what we think it did, but maybe the receptor actually phosphorylated this tyrosine. Bad news. No, it did not. And I'm not going to get too heavily into this. Dr. Larrea explained it a lot better than I could. But bottom line, guys, listen, what they did is they kept on using these experiments. They kept on using all these different tests, right, to figure out what does what and where certain things have play a role. And they worked backwards, right? So they went from, okay, S6, could it be from the receptor? Could it be S6 protein, can it be phosphorylated from the receptor? No. Okay, how about the S6 kinase? Can that be from the receptor? No. Okay, how about the S6 kinase? Can that be phosphorylated by the receptor? No. In other words, okay, so if the S6 kinase is not being phosphorylated by the receptor, that means there has to be an S6 kinase? You're telling me that there's a kinase phosphorylating a kinase that phosphorylates a kinase? Oh my God, Steven, you're killing me, man. What are you talking about? But yeah, it really is just them working backwards, guys. Okay? And at the end of the day, they came up with the hypothesis that it's activated by a cascade of events. The MAPK and ERK at the time, right? The S6 kinase, which they S6 kinase, which they named the MAPK or the ERK. ERK is just a family of MAPK. It's activated by a cascade of events. And that's exactly right? We have a cascade of events, and the activation of a class of kinases known as the MAPK, and the ERK is, just again, a member of the family. That is going to be part of a kinase cascade, and that's why we call it a pathway, right? It's a cascade of one thing leading to another, to another. So I'm not gonna go too far more deeply into the hypothesis aspect, and definitely do check out Dr. Larrea's video and the audio recording on this whole hypothesis, because it is interesting, and it does help clear things up a little bit. But I think now what I'm going to do is just jump right back into that insulin receptor and kind of try to just lay out the steps again. I know I said I would just lay out the steps in the beginning, but he really makes a great point here. So let's jump into that. But guys, just really quickly, I do want to just finish up Dr. Larrea's point here, because it is helpful, right? This is going to kind of clear up the whole nomenclature discussion that we had here, right? Okay, so we talked about MAPK and ERK, right? MAPK and ERK. When you hear these two terms, MAPK and ERK, think of them kind of interchangeably, because they are. They're going to be relatively interchangeable. ERK is simply part, as we said, of the MAPK family. All right. Now we saw that here, this MAPK-ERK situation, that was our S6 kinase. S6 kinase. And we said just now that something, we didn't know if it was the receptor or something else. Something was phosphorylating our tyrosine and threonine residues. Okay, so they wanted to check, is it this receptor? No, it wasn't. Yeah, big surprise. We probably could have guessed that. No, it wasn't. But wait, hang on a minute. Did you say threonine, Steven? I understand tyrosine. Yeah, we talked about that, but threonine? Wait, how can you have a tyrosine and a threonine being phosphorylated? I don't get that. Oh, wait a minute. Yeah. You talked about, earlier, we talked about how there are three different kinds, right? There are three different kinds of kinases that we should be familiar with. There's tyrosine kinases, there's serine threonine kinases, and oh yeah, there's those dual specific kinases. Remember? And those do both. They have two active sites that phosphorylate both tyrosine and serine threonine residues. Wait a minute. Okay, so here we have a tyrosine, here we have a threonine. So wait a minute. This must be phosphorylated by a dual specific kinase. And what do you know? That's what we see over here, guys. This MEK is a dual specific kinase because we identified that MEK, that activated MEK, I should say. That activated MEK is what is phosphorylating our tyrosine and our threonine residues. Okay, what in the world why'd they call it MEK? Well, because this is that, remember here we have, this MAPK-ERK is our S6 kinase. The kinase for the S6 kinase, in other words, the S6 kinase, stick with me, guys, that is our MEK. So that MEK, M-E-K, MAPK, ERK, kinase. Oh, wait, okay, Steve, oh my God, that's what he meant by nomenclature. That's why nomenclature is important. Because, I guess, Stephen, you were babbling before, you were mumbling, I had no idea what you were talking about. Now I see where it's all coming into play. Okay, so this S6 kinase, the kinase that is phosphorylating the MAPK, which we know is the S6 kinase. Okay, the thing phosphorylating this is MEK, M-E-K, MAPK, ERK kinase. Oh my God, wow, that makes sense. And we also know that because of these threonine and tyrosine residues, this MEK must be a dual specific. This must be a dual specific kinase. Oh my gosh, that's why you were talking about that in the beginning. That's why nomenclature matters. Okay, I see what you're saying. Remember all the things downstream here, right? Like the S6 kinase, right? And how that had only serine residues. Oh, okay, so that means this is just a serine threonine kinase, this MAPK. Okay, the S6 kinase phosphorylated the S6 protein. But where did it phosphorylate it? On the serine residues. Also, so that means this S6 kinase is also a serine threonine kinase. All right, good. So we have a bunch of serine threonine kinases. We have our tyrosine kinase up here in the receptor, right? This is our tyrosine kinase doing the trans-autophosphorylation that we talked about. Here are our little tyrosines. So we talked about tyrosine kinases. We talked about serine threonine kinases. Now we have in MAPK, we see the phosphorylation of threonines and tyrosines, meaning now whatever's doing the phosphorylation must be dual specific. And that, ladies and gentlemen, is our MEK, our MAPK ERK kinase. MEK. Okay, cool. So what's turning this on? What is turning on the MEK? What's phosphorylating the MEK? Well, they did all the fun work behind it. Bottom line, I'm going to spoiler alert, RAF gets activated. We don't know by what yet. We'll talk about that. I'm not going to go much more into it. RAF gets activated. That becomes activated, right? And that RAF, long story short, more serine threonines being phosphorylated, right? So this RAF is a serine threonine protein kinase. Another one, right? So just keep these little things in mind, right? And so what I'm going to do now, I'm going to go back to the beginning. We worked from back to front. Now what I want to do is work from front to back, from top to bottom. We work from bottom to top. We're going to go from top to bottom, and we're going to start putting these things together in a stepwise fashion, so you guys know what's up. But the reason I went into this, the reason I discussed these hypotheses is the same reason that Dr. Larrea did. Why? Because, yeah, memorization's great. It's beautiful. Yeah, you have to memorize, but it's boring, and it's not as intuitive as you'd like it to be. If you can really understand, why is it called MEK? Why is it called MEK? Oh, it's the MAPK-ERK kinase. Okay, I get it. So how did they get to the... If you can think in these intuitive terms, then you'll be able to put the big picture into play more, right? And that's what we always want to find, the big picture. How do these little, tiny little components fit into the big picture, right? Okay, you guys get it. Excellent, excellent. We saw how we use the 3 different kinds of kinases. Now we're going to talk a little bit about the other parts we discussed, like SH2, SH3, right? The sarcomology domains. How do those come into play here? What is RAS? What does RAS have to do with RAF? What does RAF have to do with MEK and MEK with MAPK and all these fun little pieces? We kind of have a small idea here. We kind of have a small idea. We're starting to put the little pieces together. We're starting to get a little bit, but, we're still a little bit iffy. That's all right. We're going to clear it up right now. Stay tuned. So, but before we even get to that, we really need to understand that we didn't actually answer the question yet, right? The question was, how do the receptors, the tyrosine kinase receptors, set off this whole cascade leading to proliferation and survival, right? We understand. We discussed already how MEK is phosphorylated is activated, and that MEK will function as a kinase for the MAPK/ERK, right? We can think of ERK as pretty much homologous to MAPK, right? Okay. We know that RAF is going to function as the kinase for MEK, right? So here's our RAF. And here it has a kinase domain on it, a place where the kinase activity happens. Okay, but what turns this on? What turns the RAF on? Well, actually, it's kind of interesting because the RAF has a somewhat similar mechanism as our receptors, as our tyrosine kinase receptors. They will come together. All right, so here's an independent monomer. Here's an independent monomer. And when they are brought together, forming this kind of a dimer complex, then they can go on to do their job of phosphorylating the MEK. But what makes this happen? What helps bring them together? What brings these two together, these two RAFs? The answer to that question is RAS. RAS. Right? Okay, so that still doesn't answer our question, because they found out that RAF is also phosphorylated on serine-threonine residues, right? And at that point, that's where I'm going to kind of bring you back to this slide. Remember this slide that we talked about earlier in the last discussion that we had? Let me go ahead and paste that here. There we go. Paste image. There we go. Okay. So remember we talked about the different types of monomeric and heterotrimeric, right? We have the heterotrimeric family. We're going to see those heterotrimers in our GPCRs, our G protein coupled receptors, the alpha beta gamma subunit. Here, RAS is going to be related. It's going to be with a monomeric. It's going to be monomeric, right? So that RAS is going to bring these RAFs together. So what's the difference between our monomeric and our heterotrimeric? That's going to simply be that monomeric is one single subunit. Heterotrimeric is going to be our alpha, beta, gamma. So think of this kind of as a light switch. Let's look at this diagram here. Think of it as a light here. Here's the inactive. Here's the active. Right? So if we want to turn it on, make it active, we bring in a GTP. We kick off the GDP. Same thing as the GPCRs, right? Right? So we have a GTP now attached to the G protein. And we'll talk about what that G protein is. If you want to take it off, you kick off, you hydrolyze that GTP to kick off 1-phosphate, and now it's GDP. Now remember though, you don't phosphorylate the GDP when you're turning it on. You don't phosphorylate the GDP to make GTP. No, you exchange it. Guanine nucleotide exchange factor. Exchange factor. Here's the GEF. Right? That's going to simply switch them out. It's going to switch out the GTP and bring in the GTP and kick off the GDP. But when you're going from active to inactive, right? then you are going to hydrolyze. You're going to hydrolyze. You're going to break the GTP into pieces. You cut off one inorganic phosphate, and you're left with GDP up here. Okay, so be aware of these differences. All right, you don't phosphorylate the GDP to get GTP, but you do break down the GTP to get GDP. All right, just a little thing to keep in mind. So what was the GEF? What was the guanine exchange factor in G protein coupled receptors? Well, it was the receptor itself, right? It was the receptor itself. So who is the GEF in our monomeric families? We'll answer that now. So to answer that question, what we're going to do, we're going to bring everything full circle. All right. Now we're gonna finally, this is where we're gonna finally answer the underlying question here. Okay. Let's start from the beginning. EGF, right? The EGF binds the ligand. We have dimerization of these two. We have trans-autophosphorylation. Now we see all these little tyrosine residues have been phosphorylated, right? They have been phosphorylated. Okay, excellent, great. Now, when these tyrosines are phosphorylated, nearby proteins recognize it. Now, let's bring back a little discussion that we had before. SH2, SH3, right? SH2, SH3. Those, we know SH2 is going to be for phosphotyrosine. SH3 is going to recognize proline-rich regions. Now, I'm going to just cut right to the point. When we have RAS, a RAS-specific pathway, that GEF that we were talking about before, that GEF specific to RAS-specific GEF, is something called GRB2-SOS. SOS standing for son of sevenless. There was a whole study on the flies and stuff, and it's just where they got the name. This complex is going to function as the guanine, the exchange factor, right? The guanine nucleotide exchange factor. So we have the phosphorylation that we talked about. Once these tyrosines have been phosphorylated, right? GRB2, the GRB2-SOS complex is going to be attracted to those phosphotyrosines. And we know, okay, so what does that mean? The SH2, here's a picture we could use. The SH2 domain, which we know is going to be attracted by phosphotyrosines, that is going to be the part binding over here. The SH2 domain of the GRB2 is going to bind to the actual receptor over here. The SH2 domain is attracted to these phosphotyrosines, right? And the SH3 domains of the GRB2 are going to be attracted to these. Here, it says P. Let's write proline, just for clarity's sake. Here, proline. I don't want you guys to think that means phosphorylated. Proline. Okay, you get the point. So these SH3s, these SH3 domains are going to be attracted to these proline-rich regions, right? So there's going to be an attraction here. There's going to be an attraction here. Okay, this whole thing is our GRB2 SOS complex, and that, ladies and gentlemen, finally is our GEF. That GEF, that guanine nucleotide exchange factor, is going to be responsible for activating what? RAS. RAS is going to do what? Bring together the two RAFs and activate them. RAF, and then RAF is going to activate our MEK, right? Our MAPK-ERK kinase. MEK is going to activate our MAPK/ERK, right? Our MAPK/ERK is going to activate all the downstream pieces. So we filled it in. We finally finished we figured out how is the receptor over here related to all of the rest of it. is through this guanine nucleotide exchange factor, GRB2 SOS. Great. So we figured it out. Now let's go through the actual steps. All right, this is the moment you've all been waiting for. We just prefaced it all. Okay, so this is now you get why. You get what we're talking about here. So let's go. So let's just bring in the whole picture. Now we're going to talk about the overall pathway, okay? I'm going to bring it back to the insulin receptor. There are a few little nuances and differences from what we just talked about. You know, I don't want you to get too bogged down with the details, right? We're going to talk about something called IRS1 over here. Don't pay too much attention to that. I don't think that's going to be a huge focus, at least from what I can tell from Dr. Larea's slides, but it's all this part here that really matters, right? The rest of it after that I want you guys to really focus on. So I'm going to just go through it for the sake of being complete, you know, being thorough, and so that you guys kind of know what's going on with these 1, 2, 3, 4, 5s, you know, all these different parts of the diagram. But you know, it's really what comes after the IRS1 that matters. So let's just start off over here, right here, and on top we have our tyrosine kinase receptor, right? And this is specifically an insulin receptor, right? INSR. We're going to call it insulin receptor. So we are going to see an insulin come in, the insulin substrate, the insulin ligand is going to just come in, bind to this receptor, right? And this is also a little bit different from the EGF that we talked about, the EGFR, but same idea, right? The binding of the substrate will cause homodimerization. Right. Once we have that homodimerization, we're going to have these two monomers come together and form a dimer. Homodimerization. At that point, we're going to see that autophosphorylation we talked about, where these individual monomers are going to phosphorylate each other. Right. Easy enough. We get that. Okay, great. So let me clean that up a little bit. We have the phosphorylation on these kinase aspects. Here's the kinase, the catalytic domain. These are kinases now, right? These are kinases. So this one will activate this one will activate this one by phosphorylating on the, on the tyrosine residues of the kinase domain. These are also tyrosine kinases, right? So they autophosphorylated. Now these kinase domains, these catalytic domains of each monomer are now active. They are now active, right? So they, being active, can now phosphorylate other things, right? So these are phosphorylated. These have been phosphorylated by each other, right? Yes. So they can go on to phosphorylate other things now. We never mentioned this IRS1 protein. We never talked about it. This is specific to insulin. When we talked about EGF, epidermal growth factor, we saw the GRB2 binding. Here's the GRB2. We saw the GRB2 binding directly to the receptor, directly to the phosphotyrosine part. This IRS1 is just a little bit of an intermediate extra step, okay? Don't get too bogged down with details. Don't freak out. Don't worry. This is just a little extra step in insulin receptors, all right? But the rest of it is what matters. But long story short, after our phosphorylation, once the insulin receptor is autophosphorylated, it becomes an active tyrosine kinase. We talked about that here, right? We talked about that here. These are your active tyrosine kinases over here. And that means that they can phosphorylate tyrosines on other proteins, right? So once they're phosphorylated on several of its tyrosine residues, this IRS1, right? So this IRS1 will come in. These kinases are going to do their job. They're going to phosphorylate the IRS1, and they're going to give you a phosphorylated IRS1. And here are all of our tyrosine residues, and on those residues we have our phosphorylation. Okay, great. Don't pay attention too much to that, guys. That's just the little middleman before we get to the meat and potatoes over here. Okay. So now that the IRS1 has been phosphorylated on several of its tyrosine residues, it becomes the point of nucleation for a complex of proteins, okay? And that's going to carry the message from the insulin receptor. Here's our insulin receptor. This IRS1 is going to carry the message that, oh, we're on now, we're active now. It's going to carry the message on to different end targets in the cytosol and eventually in the nucleus through a long series, this whole deal, of intermediate proteins, okay? So first things first, a phosphotyrosine residue on this IRS1, like the one over here, right? Here's a phosphotyrosine residue, another phosphotyrosine residue, right? These are also phosphotyrosine residues, right? With EGRF or EGF receptor, we didn't see this IRS1 middleman, but you know, these also are our phosphotyrosine receptors. Okay, these phosphotyrosine receptors, right? They are going to bind to the SH2 domain. Now we're talking, you know, this is what I need you guys to understand. This is the important part. These phosphotyrosine receptors are going to bind to the SH2. Let me go back. To the SH2 domains, which are over here. Here, different color. SH2 domains, which are right over here on the GRB2 protein. And a lot of signaling proteins are going to contain this SH2, that Src homology 2 domain, right? And we know SH2 means what? SH2 equals phosphotyrosine. Okay? So we turned on the receptor. If we turn it on, that means we have phosphotyrosine residues active, whether it be from autophosphorylation in the actual receptor, or maybe we phosphorylated something else on their tyrosine residues, like this IRS1, whatever the case may be. Either way, we have phosphotyrosine residues. Okay, now, important, now this is a key player. This SH2 domain on the GRB2 protein. The GRB2 protein. Now you got to keep this in mind. The GRB2 protein, very important. That's the growth factor receptor bound protein. I don't think you really have to know all the long names. GRB2 should be sufficient. But when you think GRB2, right here, GRB2 equals adapter. This is just an adapter protein, right? It has no intrinsic enzymatic activity. All it does is bring two different proteins together, right? So this, all we care about, all it's going to do is bring two proteins together. It's not a kinase. It has no enzymatic activity whatsoever. GRB2. So here's our GRB2. It brings two proteins together. What are those two proteins? Well, in this case, it's going to be our IRS1 and our SOS, our son of sevenless, which we just talked about. Its job is simply to connect them. It's an adapter. It's like when you have an extension cord, right? You just want to make it longer, or you want to connect two different things that are far away. You use an extension cord. This is all it is, an adapter. Okay? In order for the signal to continue on, continue going downstream, right? To continue going downstream, we need to make sure that this guy can talk to this guy. And that's the point of GRB2. Clear? Yes, I think we're good. Excellent. So, in addition to that SH2 binding domain that we talked about, here's our SH2 binding domain over here, connecting the tyrosine residues, the phosphotyrosines to the GRB2. It also has an SH3 domain. Now, who can tell me? Remember what the SH3 domain does? So SH2 is phosphotyrosine binding. SH3 is going to bind to our proline-rich regions, right? So that second protein binding domain, the SH3, will bind to the protein-rich regions of our SOS. Right? So this is going to be an SH3 domain on this side. So GRB2 to SOS is SH3. GRB2 to whatever the other protein is, right? In this case, IRS1 is SH2. Okay? So this GRB2 has two. It has SH2 and SH3 domains. Right? Dual specific. Good. Great. So now we have allowed this protein to talk to the SOS. Now, what is the SOS? The SOS is going to be that GEF, that GEF. The SOS, let me write it down here. The SOS equals our guanine nucleotide exchange factor, our GEF. The son of sevenless is going to function as our GEF. Remember, in G protein coupled receptors, that was the receptor itself, right? That was functioning as the GEF. Here we use the SOS as our GEF, catalyzing the replacement of bound GDP with GTP on the small G protein RAS, right? Before we had the alpha, beta, gamma subunits, here we have that one monomeric G protein, and that G protein is RAS. All right. So in its GTP bound active form, right? So let's say SOS did its job. Its job is it's the guanine nucleotide exchange factor is going to take what's its job here, right? It's going to take GDP and turn it into GTP. That's not going to actually turn it into that. We know that, right? It's just going to simply exchange them, right? So it's going to actually be sending GDP out and bringing GTP in, right? It's not actually phosphorylating GDP to give us GTP. It's just switching them. Exchange factor. Okay, great. So let's say it did that. Now it's active. So now we have an active form, right? At this point, the GTP bound active form of RAS, here's our RAS, right? That's our G protein. The G protein is monomeric. That RAS now has an GTP attached to it. And you see, I keep saying ATP and not GTP. You gotta be able to make that distinction, okay? I'm messing it up. Please use my mistakes to your benefit. As it's not ATP, G protein, GTP, right? G protein coupled receptor, right? GEF, guanine nucleotide exchange factor, not adenine. All right, so just keep that in mind. Now we have an activated RAS, which means what? Now we have an GTP attached to it. And you can see that over here in the diagram. Here's RAS, GTP comes in, GDP goes out. Excellent, great. So now we have in its GTP bound active form, that RAS can activate a protein kinase. Now, is RAS itself a protein kinase? No, RAS is not a protein kinase at all. RAS cannot be a protein kinase. Why? There's no intrinsic kinase activity. All it's doing is functioning as a connector, right? It's simply it's not an adapter like the GRB2 was, but it's just what's holding on to the GTP, right? It's just the G protein. Okay. That RAS is going to turn on the kinase RAF, right? It's going to activate here. Activated RAS will bind and activate RAF1 over here. Let me use a different color. How is it going to do that? Through the GTP activity. Right? But it's not actually a kinase because it's not phosphorylating. It's not phosphorylating anything on RAF at all. Okay. Now, so we said in its GTP bound active form, RAS can activate a protein kinase RAF, right? RAF1. And that's just the first of 3 protein kinases. Protein kinases. The first of 3 protein kinases, and they're all going to be in blue here. Right? Let me go ahead and get my pen. They're all going to be in blue. So MEK is one of those kinases. RAF is another one of those kinases. ERK is another kinase, right? Or we can say MAPK/ERK, right? MAPK, because remember, ERK is just part of the MAPK family. So it's the first of 3 protein kinases. RAF is the first of those 3 protein kinases that form a cascade in which each kinase activates the next by phosphorylation. Remember, we already did this, but we did it backwards. Now we're going forward. So the protein kinases MEK and ERK are activated by phosphorylation of both a threonine residue and a tyrosine residue. Okay? So here we have MEK, ERK, right? These phosphorylations are going to happen on a threonine and a tyrosine. A threonine and a tyrosine residue. Okay? That's important to remember. Where are they being phosphorylated? Okay? When activated, this ERK, right? We know it's activated because we have the phosphorylation here. This ERK will go on to do whatever it has to do. Oftentimes we'll just move right into the nucleus, phosphorylate different nuclear transcripts and different factors and transcription factors like ELK1 and all that. We didn't go too in depth over there, but right now this is mainly the aspect I need you to focus on, right? So the proteins RAF1, MEK, and ERK are members of 3 larger families. Remember how we talked about, kinase, and all the different kinases that need another kinase, and a kinase. Yeah, so let's just talk about that real quick and label what is what here. Okay? So here, we have 1. Ligand binds, causing dimerization. Dimerization leads to autophosphorylation. Autophosphorylation does what? Autophosphorylation leads to phosphate groups on the tyrosine residues, right? Okay. So we have, I'll put this, PY, phosphotyrosines. Those phosphotyrosines, right, they're going to be recognized by the SH2 domains, whether it be of IRS1, right? If sometimes there's a middleman, IRS1. But we're going to focus on the more important part. The SH2 domain of GRB1. Use a different color. GRB1's SH2 domain binds the phosphotyrosines, right? Again, GRB1's SH3 domains bind what? Proline rich regions. Okay. After that, after it's bound to the proline-rich regions, what do proline-rich regions where? Proline-rich regions on the SOS. The SOS. That SOS equals what? What is that SOS? The SOS is our guanine nucleotide exchange factor. SOS is our guanine nucleotide exchange factor, right? So we're going to take GDP out, GTP in, and feel free to just rewrite this, more neatly in your own words. But I'm just giving you the general idea here. Okay, so once you have GTP bound, we have active RAS, right? Active RAS attached to here. We have GTP bound to the RAS, meaning it's active, right? The GDP left. This is our monomeric G protein. Active RAS recruits RAF. And activates it. Activates RAF. And we talked about how it's going to bring 2 RAFs together, and similarly to the receptor, the RAFs are going to dimerize, right? RAF, or I should say active RAF with the star. Active RAF does what? That's a kinase. It's going to phosphorylate. We'll use orange here. Phosphorylate / activate MEK. right? Now we have MEK. Remember the MAPK ERK kinase. MEK, which is now active. Active MEK will phosphorylate / activate ERK, and then ERK will go on to do the rest of the cell signaling. Yes? Yes. Now, the proteins, RAF, MEK, and ERK, as we said, are the members of 3 larger families for which several nomenclatures are used, right? Nomenclature. ERK is in the MAPK family. We know that already. Let's go over here. ERK equals MAPK, right? MAPK. You're in the MAPK family. Excellent. Soon after discovery of that first MAPK enzyme, remember the hypotheses we went through, which was named the MAP kinase, right? That soon after its discovery, the enzyme was found to be activated by MAP kinase. So here's our MAPK, our MAP kinase. Next, we have MAP KK, right? MAP kinase. Let me write this out a little nicer. MAP KK. All right? MAP kinase. And what is that now? What is phosphorylating our ERK, our MAPK? Well, let's look back at our chart over here. I'll give you guys a second before I answer it for you. Yeah, MEK, right? Right? So here's our ERK. Here's our ERK. That's being phosphorylated, right, by MEK. So that means here, that MAP kinase is our MEK. Okay, but now we have MEK. What's phosphorylating MEK? Well, that's going to be our MAP kinase. And you guys know what that's going to be. What is doing the phosphorylation of MEK? RAF. RAF is our MAP kinase. Right? Or just a somewhat less cumbersome way of explaining it. You know, ERK is your MAP K, MEK is your MAP KK, RAF is your MAP KKK. Okay? Okay? Anyway, I understand it's a little bit cumbersome. It's a little bit much the first time around, and I get it. You're not alone. But believe it or not, you guys know more than you think. It might seem like a lot right now, but you know more than you think. You understand more than you think. It's just a matter of going through it a few times and really, just getting comfortable with the whole cascade. The result of this is the result of all that we discussed, of these different MAPKs, right? Of all of these different, these portions here. Let me clean that up. All of this discussion here that we talked about. The result is a cascade of reactions that amplify the initial signal by many orders of magnitude, right? That amplification we talked about, right? One little signal can yield a huge response, right? So that MAPK cascade that we talked about, that's the first of the cascades we talked about in terms of RTKs, receptor tyrosine kinases. Those MAPK cascades, like the RAF, MEK, ERK sequence that we talked about, mediate signaling initiated by a variety of different growth factors or epidermal growth factors like our EGF, right, or our insulin receptors, which are also tyrosine kinases. These are all different tyrosine kinases, guys. And, there are just different, there's little different schemes, right? So you see a little bit of a different way of the insulin receptor working versus the EGF receptor that we saw in Dr. Laraya's slides. But the point remains. The point stands that the key players, right, like the RAS, RAF, MEK, ERK, and know that ERK is homologous to MAPK. Get familiar with these terms, because I'm sure by now you understand why nomenclature was emphasized so heavily during this lecture. Okay, so go ahead, feel free to take feel free to take this little list I made on the bottom here. And I apologize, I know it's a little bit messy, but I don't have too much time to be an artiste out here drawing pictures and writing things out. But rewrite it in your own way, in your own terms. Read the textbook. Go on YouTube. Rewatch Dr. Laraya's lectures and really and try to make your own little pathway. Mine's super messy, and you know what? I'm okay with that. That works for me. Okay, I've never been an artist. I'm a student who is trying to learn the material in a way that best suits my thinking patterns. All right, and it's more than likely not going to fit yours exactly. So I implore you to figure out a way to take everything that I just told you, everything that Dr. Laraya told you, more importantly, and put it into your own words, into your own diagrams, into your own way of understanding visually, so that you can explain it to yourself later and truly understand what's happening. Okay? Now I'm going to try to go through the rest of Dr. Laraya's slides and go into some of the nuances of what he was talking about and try to just break down what was being discussed. I really wish we had that second audio recording so I know exactly where to stop and how deep exactly to get, but that's just the way it is, and that's okay, because being the resilient students that we are, we can work with whatever happens to come our way, and we'll figure out a solution regardless. Okay, so in the next few minutes, I might go a little either too deep or not deep enough. I do suggest that if you are confused, or if you're unsure of exactly how deep to take your studying, definitely reach out to Dr. Laraya. He's very receptive. He will definitely have a much more intuitive and a much more accurate, I should say, answer than I would have for you. He's the one who's going to be writing the test questions and grading your test questions, not me. Okay? Or I might be grading them, but based upon what he tells me to do. So, if you really want to maximize your understanding of how to go about your studying, reach out to Dr. Laraya directly. And he's very receptive. He's very helpful. He's spent many more hours than he's had to, helping me alone with questions I've had, whether it be with histology or even just my path in medical school or toward medical school in general. He's highly regarded in this school for a reason, and I suggest you guys not only take advantage of what information he has to offer you, but also build a bit of a rapport with him, right? Because he can really offer you some advice that I could never hope or dream to offer you when it comes to developing yourselves for, and preparing yourselves for a medical school level career. Okay, so I'm going to go through what I can over here, but definitely reach out to Dr. Laraya for things that are a little bit beyond my scope. All right. Now, I just do want to quickly clarify. I think I misspoke earlier. RAF 1. Please correct this if you wrote it down already. RAF 1 phosphorylates MEK onto serine residues. Okay, so here we're going to have a serine. Okay, 2 serine residues, and that will activate the MEK. Okay, that'll activate the MEK. Okay, and MEK will phosphorylate ERK. MEK will phosphorylate ERK on arthreonine and tyrosine residues, thus activating it. Okay, and again, I believe I did misspeak earlier, but that's okay. I'd rather I make a mistake and you guys learn from it now than you guys make your own mistakes on the exam. Okay, so use these errors, use these little things, and if I didn't misspeak before, and it's very possible because this is a pretty long lecture so far, but point it out to me so I can bring that to the attention of everybody else. Again, I was sitting in the same seat as you guys very recently, right? I do not claim to be an expert by any means in any of these things, but I did make it through. And there's no reason that, if I made it through, you guys definitely can make it through. Okay, so pay attention. If there are inconsistencies, point them out to me. Okay, I'm not, pride is not a factor here. Okay, it's a matter of getting you guys the right information. All right. Excellent. So let's move on. All right, guys, so let's move forward into the next signaling pathway, the membrane phospholipid PIP3, right? The PI3K pathway. PI3K, right? So that GRB2 that we talked about, right? That GRB2 is not the only protein associated with the phosphorylated tyrosines. We're also going to see that happening in this pathway. Okay, so remember in insulin we talked about IRS1, the insulin receptor. And do pay attention to the differences between the insulin receptor and the GEF receptor that was shown to you during lecture. because, you know, there are subtle differences, like that little intermediate IRS1 protein instead of the actual receptor itself having those phosphotyrosines. So, like I said, GRB2 is not the only protein that's going to associate with those phosphorylated tyrosines, or phosphorylated tyrosines. We have an enzyme called phosphoinositide 3 kinase, aka PI3 kinase. You can see that over here. PI3 kinase. And notice it also has this little SH2 region, right? And by now, I hope we all know what the SH2 region is for. Okay? So that PI3K, that enzyme, that phosphoinositide 3 kinase, is going to bind over here to these tyrosines here, right? The phosphorylated tyrosines is going to bind via this SH2 receptor. So here's our SH2 receptor. So let's just go from the beginning over here. Let's start from the top. Okay? Ligand binding leads to dimerization. Yes, we know this. Tyrosine kinase activation. Yes, we know this. And to autophosphorylation. Yes, we know this. Okay, here's our monomer. Yes. Ligand binds, causes dimerization. Here's our dimer, right? Dimerization is going to cause tyrosine kinase activation. And those are going to result in the autophosphorylation, right? So here's our first phosphate over here. Here's another phosphate over here. They're going to autophosphorylate. Okay. That phosphorylated receptor is going to recruit our PI3K. So now that we have this receptor that's been phosphorylated, it's going to beckon, it's going to call this PI3K. And how is that PI3K going to associate with a phosphorylated tyrosine, a phosphotyrosine, so to speak, through that SH2 receptor, or through that SH2 domain, right? So in doing so, once this SH2 domain on the PI3K associates with the phosphotyrosine receptor, the phosphotyrosine domain here, phosphotyrosine, here's the PI3K. Once it associates, this will become phosphorylated, right? So we see that over here, that phosphorylated. Now that this PI3K has been activated, right? That activated PI3K is going to catalyze this reaction. And this reaction shows us the transfer, or the transformation of PIP2 to PIP3. All right, so PIP2 to PIP3. All right, and that's going to happen by the transfer of a phosphoryl group from ATP. So this is a membrane lipid, right? A membrane lipid phosphatidylinositol. We should be relatively familiar with these terms, having done the membrane lecture before already for exam 1. So this multiply charged head of the PIP3. Now you guys are going to see over here, before I move forward, PIP2 is often referred to as PI45P2. PIP3 is referred to as PI345P3. This is just telling you where they are, where the phosphates are. Just be familiar, you know, those are the technical terms, but we can just say PIP2 and PIP3. So this charged head group of PIP3, right? Here's the PIP3, and that is charged, right? It's charged negatively, so it's a negative charge. That negatively charged head group protruding on the cytoplasmic side of the plasma membrane, that's the starting point for the whole second signaling branch involving an entirely different cascade of protein kinases. All right, so when bound to PIP3, we're going to see protein kinase B, PKB, also called AKT. So when you see AKT and PKB, they're the same thing, guys. Okay, so think PKB/AKT. So when bound to the PIP3, that PKB or AKT is phosphorylated and activated by yet another protein kinase, PDK1. Here's PDK1. Okay, so we already saw the first protein kinase, which is our PI3K. Now, the next one that we're going to see in the pathway is PDK1. PDK1. And that PDK1 is going to always be kind of associated, right, with this AKT PKB. All right, so when we see an increase in that PIP3, that increase in PIP3 is going to recruit PDK1 and our PKB, or the AKT. So it's going to recruit these two guys. And the PDK1 will activate the PKB. Right? It'll activate that AKT slash PKB. Take a second to just get that clear, okay? So we have phosphorylated receptor, right? Autophosphorylated. That's going to recruit our PI3K. Our PI3K is going to catalyze the reaction of PIP2 to PIP3. Once we have that PIP3 is going to recruit PDK1 and AKT slash PKB. And once they're recruited, that PDK1 is going to activate our PKB. All right? Easy enough. So now this activated PKB, I'm going to just use PKB moving forward. AKT is just synonymous. Just keep that in mind. That activated PKB will then phosphorylate serine or threonine residues in its target proteins, and one of which is going to be a for insulin in particular, a GSK3, the glycogen synthase kinase 3, right? And it really depends on that point which specific receptor you're talking about, right? So let's just write this out real quick. First things first, we have the dimerization, right? Or here we'll say ligand binds, which causes, I'm going to number it. Ligand binds, which causes homodimerization, which causes tyrosine kinase activation, right? In that catalytic unit, on the cytosolic catalytic unit. Autophosphorylation, we know what that is by now. That autophosphorylation is going to cause the recruitment of PI3K. So PI3K, use a different color. PI3K is going to be recruited and bind to our phosphotyrosines. Better color. Our phosphotyrosines on either the receptor or, in the case of insulin, on the IRS1, right? Whatever the phosphorylated tyrosines, wherever those are, the last piece. Okay, bind to the phosphotyrosines, and that's going to be via the SH2 domain, which we should know by now is why that is. Once that PI3K associates with the phosphotyrosines, we're going to see phosphorylation / activation of PI3K, right? Activated. We're going to put a little star here. Once we have that activation, we're going to see the transfer of PIP2 to PIP3. And that's going to happen via what? Via PI3K. Active. Okay? Now we have the PIP3, right? The PIP3. And that PIP3, which we know we got via the transfer of a phosphoryl group from an ATP, that is going to have a negatively charged, right? It's a negatively charged head group. And when bound to PIP3, protein kinase B, aka AKT, is phosphorylated and activated by yet another protein kinase. So we're going to have PIP3 recruit PIP3 recruits our second kinase, PDK1. And in doing so, that PDK1, go this way, PDK1 will activate AKT, also called PKB, right? And then from there, once that PKB has been activated, we're going to see continued activation, depending on what pathway we're looking at specifically. All right, guys. So it's definitely much easier, but do keep in mind the key components here: PI3K, PDK1. We know that active PI3K facilitates the conversion of PIP2 to PIP3. Right. And that's the main idea behind all of this. Now, guys, very importantly, remember we talked about how there's phosphatases and there's kinases and how they play an important role in the balance of this whole phosphorylation and this whole cascade, right? So when we're talking about our PIPs and those types of and that type of pathway, right, separate from the MAPK ERK pathway, we need a way to turn that off. Right, and that's where P10 comes in. We heard P10 come up a couple times, and that is basically the turn-off mechanism. Right, it's a lipid phosphatase. Okay, and it's one of the most frequently altered genes in human cancer. It's a class of tumor suppressor gene products. Okay, so we talked about how PI3K. Let's look down here. PI3K responds to the insulin signal by catalyzing, or any signal for that matter, by catalyzing the transfer of a phosphoryl group from ATP to the inositol ring. Okay. Good. So that's what we're seeing here. We're seeing our PI3K here, which was that first kinase that we talked about. Here's our PIP2. That's this whole thing. Now, by adding in an ATP, we are going to take one of those phosphates, right? And we're going to give it to the PIP2 to make our PIP3. Right? So now we have 3 phosphates, PIP3, as opposed to PIP2, which only had 2. Over here, right? So we use the ATP, we kick off an ADP. We're using the phosphate from the ATP. Excellent, great, we have our PRP3. Now, what does P10 do? P10, which is short for phosphatase and tensin homolog. You don't think you have to know that, but that's going to end the response by catalyzing the removal of the same phosphoryl group that we added. Right, and that'll be released as a PI. So if we have P10 being introduced into the system, we're going to take this phosphoryl group that we added, and we are going to kick it off. Here it is. So this equals this. This equals this. We're going to kick it off, and that's going to bring us back to our PIP2, our initial phosphatidylinositol. Okay, so just keep that in mind. That's what that's talking about. That's why we saw way up before when I said that P10 seems to be blocking or turning off, right? We saw that P10 with this little symbol, right, next to the PI3K. So we're essentially saying that P10 turns off PI3K. This is usually a sign for inhibits, right? And if we see an arrow, so PI3K, we would see it take PIP2 to PIP3, right? Arrows mean it's moving forward. If you see this little symbol over here, that means usually stopping, inhibiting. All right, that's the point of P10. P10 is basically reversing the actions of PI3K, the kinase. It's a phosphatase. All right, that's the key to know here. It's a phosphatase. All right, so we're pretty much wrapping up around here. Before we talk about crosstalk between receptor signaling systems and receptor desensitization and whatnot, let's just really quickly run through the JAK-STAT pathway. Very, very simple compared to what we were just doing. JAK-STAT, here's the canonical pathway, right? Canonical, right? The conventional pathway. The typical pathway, the canon, right? So I'm going to just go step by step here because it really is pretty straightforward. Step 1, ligand binds. Okay, ligand and binding causes what? Yep, nothing new here, receptor dimerization. Okay, so we're going to see these receptors dimerize. They come nice and close together. We see these JAKs over here. That dimerization causes an activation of the associated JAK. Okay, so now these have been activated because this has been dimerized. Not the greatest picture because these kind of look dimerized already, but you know, I guess this is just more dimerized. If you look at the JAK's positioning, you can see they're kind of different. Anyway, that dimerization causes an activation of the associated JAK, which we just did. We started those, which in turn phosphorylates the receptor. Okay, that's new. Okay, so we see that over here. We see that over here. These little phosphoryl groups being added, right? So these receptors, this is the receptor, the JAK, now activated, which means it's been phosphorylated, right? Autophosphorylation. Those activated JAKs are going to phosphorylate the actual receptors, yes. And once these have been phosphorylated, STAT will be recruited. STAT will be recruited to the receptor, and once it's recruited, over here, the next step, it is phosphorylated itself. The STAT is phosphorylated, and we're going to see one on each side, on each monomer of the dimer. And those STATs, step 4, the STAT proteins will dimerize. So these STATs are going to come on down here, dimerize, and translocate to the nucleus, which is going to activate gene expression, right? So once they jump onto the double helix over here, they are going to have an influence on the genetic expression being exhibited by the cell. All right, simple enough. We already talked about JAK, Janus kinase, what it stands for. So just, you know, this is relatively straightforward compared to everything that we did before that. So if you got everything before that, this should be a piece of cake. Okay. All right, guys. Let's finish up our discussion on these pathways with a talk about crosstalk. We're going to talk about crosstalk, and we're going to talk about desensitization. Okay, so this is a nice image from the textbook that I think kind of outlines what we're talking about pretty nicely. So for simplicity, we've treated these individual signaling pathways as separate sequences of events, right, leading to separate metabolic results. But in fact, there is an extensive crosstalk among these signaling systems. The regulatory circuitry, right, that governs metabolism, it's very interwoven. It's very multi-layered. And we discussed, like, for GPCRs, the textbook talks about epinephrine, right? It talks about the epinephrine, the adrenergic, beta-2 adrenergic receptors, right? And I do encourage you to take a look at the textbook as well, and get familiar with those types of pathways too. But we talked about epinephrine there, and we talked about insulin here in the tyrosine receptor kinases. Now, we talked about them separately, but they don't operate independently. They are interwoven. Insulin opposes the metabolic effect of epinephrine in most tissues. And activation of the insulin signaling pathway will directly reduce signaling through the beta-adrenergic pathway, through the epinephrine system. So, for example, the INSR that we talked about, the INSR kinase, right, the insulin receptor kinase that directly phosphorylates to tyrosine residues in the cytoplasmic tail of beta-adrenergic receptors and PKB, activated by insulin, will phosphorylate serine residues in the same region. The phosphorylation of those 4 residues will trigger what's called a clathrin-aided internalization of beta-2 adrenergic receptors. And that's going to lower the cell's sensitivity to epinephrine. Okay, it's kind of a crosstalk. Let me try to reword that. So, here's our insulin receptor. Here's our insulin receptor. Right? Here's our GPCR. Right? Beta-adrenergic receptor, which is a GPCR. So let's focus on these two. When the insulin receptor is activated by insulin binding, here's insulin, it binds, now we're activated. It's tyrosine kinase, these kinases, the catalytic portion, right? It'll directly phosphorylate over here. This tyrosine kinase will phosphorylate parts of the G protein coupled receptor on the tyrosines, right? It's a tyrosine kinase, so it'll phosphorylate these tyrosines that we see on the cytoplasmic tail of a GPCR, in this case, the beta-adrenergic receptor. Okay. And that's going to be the carboxy terminus, just for you guys to be familiar. The carboxy terminus is going to be the inside. And at the same time, we talked about how it activates PKB, right? We talked just now about how it can activate PKB indirectly. So this is a whole different way. Indirectly through this pathway, right? We activate PKB, and that PKB is going to activate serine residues on the same tail of the GPCR, the beta-adrenergic receptor. Now, the effect of this phosphorylation, when we see that all of these guys are phosphorylated, right? The tyrosines and the serines, what's the result? The result is internalization. The GPCR will be internalized. It'll be removed from this membrane, from being active, from being usable, from being useful on the membrane. It's going to it'll get internalized. GPCR internalization reduces GPCR responsiveness. So now we have fewer beta-adrenergic receptors because insulin, which directly antagonizes beta-adrenergic activity, because insulin receptors have been activated, now that's how we control the opposite pathway from taking place. So this is just an example, but this is the idea, right? So when we see insulin be activated, which is one pathway, then opposite pathway to beta-adrenergic receptors, we're going to see one of them get internalized and rendered ultimately useless, right? So this becomes garbage. Now this is no longer useful. We have fewer receptors on the membrane to be targeted by epinephrine. So epinephrine in normal cases would come in and stimulate the GPCR, the beta-adrenergic receptor. But in the presence of insulin, which directly opposes epinephrine, right? In the presence of insulin, which directly opposes epinephrine, we're going to see this internalization take place. Okay? I hope that makes sense. It actually makes a lot of good sense in the textbook. I definitely implore you to go back and take a look at that. And of course, reach out to Dr. Larrea and see what his explanation was. I really wish I had the audio recording, but that would make things a lot easier. But I'm just trying to go by my understanding and by what the textbook is saying here. Okay? Now, alternatively, this can happen, in the other way. This can go the other way. So if the insulin receptor catalyzed phosphorylation of a GPCR on the terminal tyrosine creates the point of nucleation for activating the MAPK cascade, right? On the other side, if we look over here, on this side, here's a GPCR, right? Sometimes it can be used. Sometimes this GPCR can be used to increase. It can be used synergistically to increase the effectiveness of the insulin receptor being activated, right? So, yeah, we'd see this pathway happening normally through the tyrosine receptor kinase, but it'll also have a phosphorylating effect on the GPCR's carboxy terminus as well. And that GPCR will also carry out the same pathway. This is crosstalk. So you can see an upregulation or a downregulation depending on what's being stimulated. And that's kind of the idea behind crosstalk here, right? Extensive interconnections among signaling pathways will allow for the integration and fine-tuning of multiple hormonal effects. Yes? So I think that's what Dr. Larey is trying to get across here. I would, again, encourage you to definitely look into it a little bit more on your own and get a better understanding of what I'm talking about with regard to these epinephrine and insulin pathways. They are separate but related. Okay, guys? And I think that covers us for the cell signaling portion. Of course, feel free to ask me questions and reach out. I'll definitely try to get some practice questions out there and some clarification where needed. But for now, I think this is a good start. All right? Thanks for listening. Next up, we're going to talk about blood coag. I think we're going to do a separate video for that.